cram them all in one place. There are differences in ion concentrations inside versus outside our cells. Certain cell types, like neurons that send messages in your nerves, need it to be that way to do their jobs. So we have to use active transport to push sodium out of the cells and potassium into the cells to keep the conditions right for those cells to function. We also use some active transport to absorb substances in the digestive tract. After you digest your food, you break it down, you want to get all the protein and carbohydrates out of that you can, because our bodies are adapted to not always being able to get a regular supply of food. Agriculture is only about 10,000 years old. That hasn't been long enough for us to adjust and really having it at the levels where most of us never have to worry about being hungry is still only in some countries and even sometimes of the year. So we have to spend a little bit of energy to make sure we get all that glucose in, but it's worth it because we get so much more from the glucose. This animation, the illustration here is moving hydrogen ions, which is how we make our stomach acid. The animation is something called the sodium-potassium pump, which controls those gradients of sodium and potassium inside and outside the cell.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. 1 phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. In other words, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
Questions on active transport? Our last membrane crossing method is vesicular transport. So this is also active. The cell has to spend energy. Vesicular transport is for larger things like proteins or bacteria. If you have a white blood cell that's going to eat one, keep it from making you sick. It can also be used for larger amounts of smaller things like a bubble of fluid. The vesicle either forms from or joins with the membrane to move substances across. So you would get something like this, where the vesicle connects to the membrane, and suddenly its contents are outside the cell. And it can do it the other way around, too. The reason it needs energy is you're folding the membrane, and that takes work. The vesicular transport has several subtypes. They're all named something cytosis, and they're differentiated based on which way a substance is moving. If it's going into the cell, it's called endocytosis. Endo sounds like into. If the substance is leaving the cell, it's called exocytosis, like exit. or like you want to get your ex out of the house. Transcytosis, trans means across. Here, the substance enters the cell, moves across, and then exits it again without being used by the cell. And that's basically endocytosis, move the vesicle, and then do exocytosis. Endocytosis, we have a couple more specific varieties of it. Phagocytosis, or cell eating, is used for large particles. You may have at some point seen a video of an amoeba doing this. They kind of reach around something, and when the ends of the membrane touch, they connect in a different way to make a vesicle with the substance inside it. Pinocytosis means cell drinking. This is when it just takes in a bubble of fluid from outside. Cells might do this if their volume is getting low. But the concentration's the same, so the water's not going to move. So they go, okay, I'm just going to take some fluid from outside, call it good. There's also receptor-mediated endocytosis used for taking in specific substances. In phagocytosis, after we take in this large particle, we have to break it down. So that vesicle will fuse with a lysosome to get the enzymes to break it down. So, say your white blood cell eats bacteria and then digests it. So this can also be done if you need to clean up some dead cells that are outside the cell. Because lysosomes can only digest things inside the cell. So you need somebody to get the pieces inside them in order to break them down.
When that does that, that's what makes your temperature go up, right?
Not quite. There are a couple of different immune cells involved in fighting an infection, and some of them secrete things that go to your hypothalamus that controls your body temperature, and they say, hey, crank it up. And they do that because they can work better when it's hotter.
Gotcha.
But it also kind of weakens the invaders, so it's kind of a home field advantage. So here's illustration of phagocytosis at the top, phagocytosis itself, taking the particle in at the bottom, digesting it. Anything that's useful gets sent out of the vesicle and into the cytoplasm for the cell to use, like amino acids, so it can build its proteins. And if there are wastes that it doesn't want to use, it'll get rid of them by exocytosis, which we'll look at coming up. Other questions on phagocytosis? Pinocytosis looks about the same, except there's nothing large being taken in. And what will happen after this is the cell will just break down that vesicle, so the fluid becomes part of its cytoplasm. Then receptor-mediated endocytosis. You have receptors on the cell that are looking for some specific item. When enough of them bind, then it'll pinch off the vesicle with the receptors in its wall. And the substances in it. One example of this is cholesterol. It's transported in our blood in these big old protein complexes called low-density lipoprotein, or LDL. And instead of releasing the cholesterol for the cell just uses receptor-mediated endocytosis to take in the whole particle. And then what happens after the endocytosis depends on the substance and what we want to do with it. Usually, we have to take it off of the receptor and get it into the cell. And then often the receptors will be sent back to the membrane so they can pick up more. So here's receptor-mediated endocytosis. Our receptors fit only these pink substances. They're ignoring the blue and yellow ones. And the vesicle doesn't form until we have enough on the receptors to be worth making the trip. That might be like a carnival ride where you have to have the weight balanced, so you need enough people in the right spots before you can run it. Instead of just, oh, one person's ready and there's not a line, so let's go ahead. And we can't do it until we've got enough. Exocytosis, we have the substance in a vesicle inside the cell. The vesicle fuses with the membrane, and that opens up to the outside, releasing the substance. We saw this on the phagocytosis illustration for waste excretion. It's also the major way that cells secrete things that they produce that are meant to be used outside the cell, like proteins and mucus. So we're starting with producing a protein on the rough ER. It gets sent in a vesicle to the Golgi apparatus, and then the Golgi sends it off in another vesicle to the cell membrane. It joins and opens up, and then the protein is released. The vesicle itself becomes part of the membrane. But then if that membrane later does a form of endocytosis, the vesicle comes back in, probably a new vesicle. So, transcytosis just combines some of the processes we've already seen. We do endocytosis on one side, move the vesicle across, do exocytosis. So some purposes for this. This is for getting things across a layer of cells to other cells. For example, absorbing whole proteins in the digestive tract. This is usually for the immune system. We send them across to immune cells that are here. They need to see the protein. This is especially, a lot more of this happens in babies to take in antibodies from mom's milk, which are proteins that protect against infection. They don't work if you digest them. They have to be taken into your blood completely. Some nutrient delivery from capillaries is also done by transcytosis. If something bigger or some specific things, rather than just whatever happens to be going through here and is small enough, needs to be sent through. Then the cells in the wall will take it in and pass it through. So this animation puts together all types of vesicular transport.
The substances taken in by single-celled organisms are often particles or large polar molecules that cannot cross the hydrophobic plasma membrane. Many single-cell eukaryotes employ